Okay, so let's get cooking with chapter 14, and then we should have plenty of time for chapter 25. Pretty much what we went over in the review, for those of you who attended the review or have watched the review for Unit 2, pretty much what we went over for chapter 25 is what you would need to know for chapter 25. I don't have a lot that I'm going to be pulling from there. So chapter 14, it's mostly terms, really, more than anything. There's not a whole lot I can do to help you out there, but we're going to go through it. See. All right, so epidemiology is the field of medicine studying disease and adverse occurrences experienced by populations. It doesn't always have to be infectious disease either. So they might do, looking at epidemiology of cancer and all sorts of stuff to try to find correlations between certain things. So they're going to look at things in terms of the who, what, where, when, and why of whatever situation they're looking at. So starting with some history, these are some pretty great stories that I feel like are worth knowing. Florence Nightingale, have you ever heard that name before? But Florence Nightingale, she was a nurse in the Crimean War, and she saw that 32 out of every 100 soldiers were dying in field hospitals, okay? Not from gunshot wounds or the wounds that they came in with, but in fact from dysentery, usually because of improper hand washing procedures and that sort of stuff. And their immune systems are already weak just because they're suffering from wounds. So she instituted some changes that improved the unsanitary conditions, like cleaning beds after each patient and washing hands, stuff that we consider to be basic now, but back then was kind of revolutionary, reducing deaths to about 2 out of every 100 from dysentery. Next, we have John Snow, very well known for his effect on a cholera outbreak. They consider him to be kind of the father of epidemiology. But basically, there was a cholera epidemic in London, and people were just kept getting sick from it and kept getting sick from it. So what he did is he plotted out on a map all of the people who had cholera, basically. And he found that they were centered around a specific well, like a water pump. And they went and found out that that well did in fact have cholera, and it was just being recontaminated by people who were just going back and using it who had cholera, and so on and so forth. So he notified people that they needed to stop using that well. Of course, nobody listens. So he just took the handle off the pump so that they couldn't use it. So he actually helped stop that epidemic by recognizing that based on that map. So these are like these darker areas where they had seen cholera, patients with cholera anyways. Also around this middle area. Somewhere in there is the well, the water pump. But yeah, this is a picture kind of depicting how death, cholera is waiting at each of these. I don't know how much you guys know about cholera other than, or even dysentery, other than, if you've ever played or seen Oregon Trail, but that's how I learned about it first. But cholera is a very serious disease. It causes what they call rice water stools, and it looks like rice water, like you don't have poop in it anymore. It's just your water being expelled out of your body, all of your water, all of your electrolytes. And people who die, they're going to die from, and it's not uncommon to die from it, just to be clear. They're going to die from dehydration, Typically, there's nothing they can do to treat it other than just give fluids. And so they have these beds that they call cholera beds, and they are just like a cot, basically, with a hole under the butt area and then a bucket underneath, and just let people wait it out. And that's what you do. That's a real thing. So yeah. That's cholera. Like, it's actually a pretty serious disease. There are vaccines available for it in places that are endemic for it, which endemic, in case you don't know, we're going to learn what that term is today. All right, here are some terms. There are actually 4 terms on here, but 3 of them I can see, being bolded, but we'll get to the 4th one in a moment. Prevalence of disease. So prevalence is going to be the total number with respect to the entire population. So then we have incidence. So I usually try to think of incidence. So this is going to be new cases over a certain time period. So clearly like a shorter look at something, a smaller look at something, right? So I think of it as an incident as being a small individual thing, whereas prevalence or something being prevalent is like, you know, a bigger idea. So that's how I remember the difference between those. So it's total, prevalence, incidence, cases in a certain time period, like how many cases popped up in the past month, stuff like that. That's incidence. The other term is related to that. So our term that isn't folded is morbidity. The morbidity rate of a disease is the same thing as the incidence. So the occurrence over a specific time period. And then we have mortality rate, just like it sounds like this is going to be dealing with the deaths in the population due to that disease. All right. We can look at, so that here are, they're saying the epidemic curve patterns that it's showing here. But what I really want you to be focusing on here aren't really the curves, because I'm not going to ask you over the curves. What I'm going to be asking you about are the types of epidemics, like basically how they originated. Okay. So point source and then common source, and then, and then the other one is going to be propagated. So there's three. Okay. So the first one is point source. We're going to get broader as we go. Okay, so point source, the infectious agents came from a single source and all victims were exposed at that same time. So there's one exposure, everybody was exposed at that one time, right? So if someone were to come in here and they had a new form, a new strain of COVID that nobody in here had seen before, and they were coughing and sniffling and whatever and get us all sick, that would be a point source epidemic because we got it from that one person. We can pinpoint it. We all got sick at that same time, right? Whereas the next one, common source epidemic. So point source, a specific point, right? We can pinpoint exactly that one day, everybody got sick. Common source, it's exposure to a single source, but usually can be occurring over a period of time. And so the good example of that would be that cholera epidemic that we were talking about at that water pump. So people were going there, you know, over a long period of time. They weren't all exposed at one time, right? But it was one source. So that's a common source, but it's over a longer period of time. Yeah, and then propagated is actually pretty straightforward. When you think of the flu and how we have flu season, and we're going to have more likely to have the flu in like the winter months and all that, but it's kind of around all the time. That's propagated. So it's just kind of always there. Lately, we could say that about COVID. Yeah, it's kind of evolved into that sort of situation. But yeah. All right, so we have the index case. This is the first person that was found to be ill with whatever disease we're looking at. So that doesn't necessarily have to be the source, right? It's just the first person that led you to finding out, you know, who was sick. We'll watch something like Outbreak and what is it, Patrick Dempsey, McDreamy, right? McDreamy is in that movie, in case you didn't know. He's the first one that they find as being sick from the outbreak of Ebola, basically, but he wasn't necessarily the source. The monkey was, and he was smuggling a monkey from Africa across the ocean, and the monkey had Ebola. So that's one way to look at it. The first patient to found to have the disease, the index case. So endemic means it's going to be a relatively steady frequency over a long period of time. If you found out somebody in here had the flu, would you be surprised? No, because we see it all the time here in Oklahoma, right? Like, so it's pretty much endemic all over the US. So yeah, anything like that that you would look at being a disease that would not be surprising to find in that area, you could probably call it endemic. Okay, so you are, it's gonna be relatively steady frequency, predictable amount of cases that you would see, unless there's a new strain, right? That's a different situation in a particular area. So then sporadic disease, these are occasional cases reported at irregular intervals in random places. Good example of a sporadic disease would be if somebody were bitten by a rabid raccoon and got rabies, right? So that doesn't happen all the time. You don't see that everywhere, and you know, it'd be random, and those, you know, define cases. If somebody were to bring over super strange tropical disease into the room, you know, that we had never seen before here in Oklahoma, that would probably be a sporadic disease as long as it wasn't maintained thereafter. That's the idea. So sort of popped up, not normally seen there. All right, so, and then we have epidemics. These are whenever we have the prevalence of the endemic, so the one that was normally seen, right? So anytime your prevalence, so the like basically how many people are getting sick from the disease increases out of what you were expecting, or you have a sporadic disease that keeps going and it's no longer just sporadic, right? It's carrying on. So this is an unexpected circumstance. If we had it, like, for example, a new strain of the flu come out, that would be probably an epidemic that would be resulting from that, because it's not going to be giving you the endemic results that you're used to seeing, right? The number of people you would be seeing sick is now going to be skyrocketing with the new strain. And then it's the sporadic disease. If that turns into, somebody did bring in, you know, Ebola or something in here, it's one thing if it ends in this room, but what if somebody brought it out of the room and it continues to spread outside, right? So that would be an epidemic at that point, because we're spreading beyond what you would expect in that situation. There are numbers that you have to qualify for in order to actually be called an epidemic. So you can't just say like, oh, more people than usual have this. This is an epidemic. There's actual qualifying numbers that you don't need to know, but there are. So there's, because I don't know if you know this, but epidemiology isn't so much about science as much as it is about statistics, which is math. And I don't like math. So we're not doing it. So next we have pandemic, which pandemic doesn't mean it is all over the world. What it means is it had spread across a continent. So from at least one continent to one other continent, that would make it a pandemic. Okay. That's the only qualifying factor there. Epidemiologists think of the 4 different stages of a disease. Basically, the history of a disease is what this is talking about, the history of any disease, not over like looking back in like the 1800s or something, but literally the history of that kind of disease as we're looking at. So we have somebody who is a susceptible host who isn't infected, right? You can get infected with this disease. You're capable of being infected with the disease, but you aren't infected yet. And you have an exposure. Then after that exposure, you will start having what they call, what we should know by now, right? Be familiar with this term, pathological changes, right? So those are going to be symptoms and signs. So those pathological changes going to be seen as symptoms and signs as they progress. You know, at first they'll be small and you won't see them. They have to progress to that point, of course, obviously, because that's just like time, what happens. But you get infected here. It's growing as a disease. You have your signs and symptoms. You have your stage of your clinical disease. That's usually whenever you're going to get diagnosed because you finally decide to go to the doctor or whatever. That's kind of that acute phase where we're looking at like how diseases progress. Remember, we had talked about incubation, prodromal, acute phase, and then convalescence. So acute phase, the stage of clinical disease, and then recovery, disability, or death. Epidemiologists are pretty morbid, I'm not gonna lie. So because that's kind of their job, right? They want to prevent people from dying, but recovery, disability, or death. Those are your 3 choices if you get a disease. You have to do one or the other. So one or the other, one or the other, or the other. All right, we're not gonna go through these because I kind of already talked about this. I don't need to go into too much detail. But yeah, remember, you're gonna be susceptible and you're gonna get the disease. It has to kind of grow for a while. That's that incubation period. You start having, during the prodrome, your first symptoms. Your signs and symptoms, of course, those are different things, yes. And then whether or not you're gonna recover, have a disability or death. Remember, disability usually is going to be determined by remaining symptoms or signs, right, of a disease after you have cleared the infection. So we have a whole other word for that we learned in the other chapter, which was sequelae, right? So it has, or sequelae, however you want to look at it. But yeah, same difference. That is what that's talking about, okay? All right, detecting diseases, you want to detect them as early as possible, as soon as possible, as quickly as possible, as easily as possible. That way you can just crank out the test as much as you can. We know this because we lived through this with COVID. It went from being a brand new disease that everybody was scared about and not being able to figure out to having home tests. Yes, they were hard to find for a while, but at least we had them. And there was that idea that, okay, now we can start testing people faster and figuring out who's sick, who isn't sick, who needs to be treated in quarantine and all this and how we're going to deal with that. So screening tests are different from diagnostic tests. Diagnostic tests are usually more invasive. Okay, so the diagnostic ones, those are the ones. Like screening, think of screening as being like the home tests, yeah? Super easy, quick, but they're not super accurate, right? So if you had a positive test on your home test, nowadays you'd probably just be like, that's enough, you know, it's probably positive for COVID, whatever. But in the beginning, you would for sure go to your doctor and want to get your PCR test done to confirm that you had COVID. So the PCR test would be your diagnostic test in that case, okay? Because it's more accurate. It says more invasive with more hazards and less patient acceptance. It's hard to think about it that way as far as like with COVID as our example, but they give it a better example here in a moment, which will make a little bit better sense there. Yes. So the screening tests, we want them to be non-invasive, cheap, easy to perform, even perform at home. We can even deploy them on healthy populations to detect diseases before it causes harm within that population, so we can isolate infected individuals before it spreads further. Positive screening test is usually going to be followed by the diagnostic style test. It's a little bit different, like we said, with COVID, just because it's so prevalent in the society now. But the good example that they have here, a pap smear would be considered a screening test. You know, they give a brushing of the cervix. It's not super comfortable, but that's all it is. If there's something unusual with that, it's not going to tell you too much about the details of what's going on with that individual. So they'll call them in for a biopsy. You usually start with a colposcopy, which is a little pinch of the flesh, and then send that in for pathology. But yeah, so that's going to be more invasive. And it's a lot more painful to get the biopsy done, even with the little numbing gel they put on you. All right, specificity and sensitivity. So this is the one that's going to trip people up. Okay. Let's just go through them. Specificity is a term that is determined by how many people it's going to call positive who aren't. So this is determined by the false positives. Okay. How specific is it? The more specific it is, the less false positives, okay? It's specific because it's not just going to call it willy-nilly everything positive accidentally, broadly, just an attempt to, you know, call it all. It wants to be only calling what's positive, and not other things positive, okay? So that's what this is concerned with, specificity. Sensitivity, determined by how many people who were positive, but we called them negative. Those are false negatives, okay? So you want insensitivity to reduce the false negatives because you want to pick up everybody who could possibly be positive. You want to pick up every single positive there is. So ideally, you can have both. They sound like they're mutually exclusive. They're absolutely not mutually exclusive. You want to reduce your false positives and your false negatives, right? So that's the idea here. You want it to be both specific and sensitive. The way that to me, sensitivity makes more sense, right? So that's how I think of it. As long as you can remember the two words, specificity and sensitivity. Sensitivity, like says how many people were positive, but they were found as negative. So you want to be as sensitive as possible. You want to pick up as many of the positives that are out there. You want to find every positive there is. If there is a positive, you better be popping positive, right? So it's very sensitive because it'll pick up anybody that's like even slightly positive. That's the idea of sensitivity. That usually makes more sense to people. So if you want to focus on that one and just think other one is specificity, then that is fine, as long as you remember what the term is, right? Because there could be a choice of a whole bunch of S words and you'll have to pick out one of them. But yeah, so that's how I try to remember the differences between those, but whatever you need to do. All right, causality. We've already talked about Koch's postulates as far as taking samples from a person, isolating the organism, putting it in another animal. It has the same disease, and then re-isolating that organism again. That's how he discovered what was causing anthrax. We apply that same ideology now, but using molecular, testing techniques. So like PCR and stuff like that. But we have to be very careful, because correlation does not mean causation. And I don't have a good example for this to explain this to people, but anybody that knows what I'm talking about knows exactly what I'm talking about here. Because just because somebody was found at a crime scene and was the only person there and has blood on their hands, doesn't necessarily mean that they were the killer, right? You need to do investigation to prove that they were or weren't the killer. Let's just say they found the body there and they were just trying to help them out, and they really were just trying to help. Like, you don't want to just put that person in prison just because they were there with blood on their hands. So that's correlation. Then, actually determining the cause, you have to determine, like, do all this testing to prove that it's not just a correlation that that person had the blood on their hands, that they were actually the one that pulled the trigger or whatever, right? So that's the idea. That's the difference, and let's not get too confused about it. Whenever we get into talking about some of those diseases, people forget that correlation is not caused, especially on the news. The news does not know the difference between correlation and causation. I think we can all agree on that. But anyways, so we can perform experiments. Of course we can, to try to determine, what's going on. We have these randomized control trials that are very much related to what you would see with drug trials. Okay, typically you're going to have experimental group and the control group, right? So control group getting the placebo and the experimental group getting the drug, and neither the doctors nor the patients know who is getting what. And then they will look at that data afterwards, after the study, to determine, that sort of stuff. So that's the idea with a randomized control trial. You want to take out any other sort of thing that could interfere with your experiment. I'm not going to ask you about randomized control trials, by the way. I will ask you about these. Okay, and I said if you were watching on I know most people weren't on the review, even though we had quite a pretty good turnout. We had like 34 people, but I have like 140 students. So, so it's not actually all that many, but I'm not judging, believe me. So we have these types of studies: the cross-sectional, the case control, and the cohort studies. Those are our 3 studies that we can do as epidemiologists to figure out causation. Okay. There is going to be a test on your exam. There's going to be a question on your exam about, I'm going to give you a scenario, and it's going to explain to you this is what's going on, and what kind of study is going on will be your question, basically, at the end of it. Okay. So you'll have to tell me, based on the information that I'm giving you, whether it's cross-sectional, case control, or cohort. There's a 4th choice too that's meant to trip you up, of course, to get you thinking you need to know these terms, be able to recognize them at least. So you'll have to know which ones those are. That is definitely the most difficult question on the test. And it's the only critical thinking question on the test. So don't panic too much, because there's not a lot of them on there. It is the only critical thinking. The rest of them are all questions like, I think what you would expect, like what you see on the quizzes and the Kahoots and stuff like that, just basic questions, right? Yeah. I feel like this test is more forgiving, and obviously there's a reason for that, because the content is not, right? It's difficult content, so I try to make the questions a little bit more forgiving. But anyways, like I said, people usually do better on this test. It's usually because I usually get easier questions than the first one. The first one is to get you guys thinking about the science behind it and actually using that stuff in your minds. People come with a clearer head into the second one as well. So cross-sectional studies anyways, there are 2 factors, let's say exposure and disease, okay? What the disease is and where the exposure was that occurred in this population. They're collected at a single time point with no reference to which one may have occurred first, okay? That's basically how we would look at 2 factors, single point in time, don't know which one occurred first. You're just collecting the data irrespective of that, okay? Case control studies, a group of people will be categorized whether they have the condition or not. Those groups are asked about their exposures, and then we're going to see whether a certain exposure stands out in the group that is sick, that had the disease, okay? So that means like taking everybody in, the whole of science building right now, and taking the ones who have the flu or something and separating them from the ones that don't. And then being like, okay, what did you do in the last, week or something? And then making note of all the things that these people did, literally all of them, in the last week, and seeing if there's any sort of matchup there. And that would be probably more or less still a correlation. But then you have somewhere to start to go look for other data in place for where that outbreak is coming from. So yeah. All right. And then we have cohort studies. These are people that don't have the illness. They just don't have it. And they're entering in this study, and then they will just be monitored over time for the disease. And then they're going to look at exposure factors for those people who get the disease during the study. So they can actually watch somebody from start to finish if they do happen to get the disease, where they were, what they were exposed to, and all that. They just say, hey, just keep a track of, where you go basically in the next month or two, or when you get sick, let us know. And then we're going to look at your data. That's basically what a cohort study is. This one to me makes a lot of sense. This one, case control study, makes a lot of sense. This one up here doesn't make a lot of sense to me. But, I guess looking at the two factors at a single point in time, and I don't know, whatever. I don't know. But the other two make more sense to me. Okay, so moving on. I mean, just saying, in case you're not picking up on what I'm trying to throw at you, if I don't understand this well enough, it's not going to be the answer. Okay, I'm not going to make that the story that I wrote for you. Okay, if I don't understand it well enough, I'm not going to ask you the question about that difficult question. Okay, I'm not going to ask you anything beyond like just basic definition. Surveillance, basically looking at, I don't know what it sounds like, the mortality, the morbidity, the transmission, the occurrence, everything going on with a disease at any point in time. You want to keep track of that stuff for certain diseases, right? So if we're having, a new strain of COVID come out, then you want to start looking at everything related to that, so you can keep track of whether or not you think it's going to be a more severe, epidemic than with the other strains, or how many people you're likely to see die as a result from this disease, and all of that stuff. So that's the idea with surveillance. That's all it means. It's just watching out to see what's going on with that. So we have notifiable diseases and reportable diseases, same thing. The government might require you to report, whether or not a patient is diagnosed with a certain disease. These will be things that are usually highly contagious, highly dangerous, or highly unusual, especially for that area. And they're usually required by law. Some of these federal law. So if you had a patient that came in the beginning with COVID, you had to report it by law to the CDC, because they're trying to keep track of everything. So you can't keep track of it if you don't tell people, that your patients, because some people will be like, oh, well, they're tracking these many, this many percentage of the population has COVID. How do you think they get that data, y'all? They got to report it. Like, it doesn't magically appear. Your doctors don't just shoot that data out into the universe for people to have. So they have to report that. It's usually two places, like the CDC or the local health department, right? Not all diseases have to be reported to the CDC or the World Health Organization. Some of them might just be local report. If in Oklahoma City, we're more concerned about whatever strain of blah, blah, blah than other areas in the United States, then our local government might want to be tracking that. This is, of course, going to be things that are going to be very concerning disease. Things like if somebody came down with Ebola, you would want to report that to the CDC, for example. But okay, we should have said this. So CDC comes out with a report every week called the Morbidity and Mortality Report. So how many people are getting sick and how many people are dying from diseases that are of concern. So just to keep track, and so everybody who's interested as far as the epidemiology of these diseases can keep track. This is also primarily what news stations are going to be referring to as well. So this is public information. It's not anything secret. Right, healthcare associated infections. This is, of course, a thing. If you don't know this, if we talked about C. diff enough, you probably are aware that that's not something that just happens around normal populations as much. It's going to be healthcare associated. So you're going to be somebody who's usually staying in a healthcare facility. Not always hospitals. It could just be long-term care facilities, and it could be things like nursing homes and that sort of stuff. So you actually see a lot more of these in nursing home residents. Some of these, at least. And this 1 in 31 hospital patients and only 1 in 43 nursing home residents has a HAI. So I don't know if that is necessarily true or if that's what's being reported, So anybody who's worked in a nursing home probably understands what I'm talking about. So people don't always let people know about what's going on with their patients, unfortunately. But yeah, so it's bad, and we all know it's bad, and we all know that these are the things that are typically associated with that. Catheter-associated urinary tract infections, obvious. Central line associated bloodstream infections, another one that's obvious, right? If you have a long-term central line, or if somebody's not cleaning the area properly before they start their central line, there's gonna be problems. Surgical site infections and ventilator-associated events. So this doesn't always have to mean ventilator-associated events. Doesn't mean necessarily infection. It can. People can get pneumonia associated with being on a ventilator. Very common, actually, but it could just be other problems related to being on a ventilator, structurally, physically, that sort of stuff. All right, how do we prevent these things from happening and keep it under control? Well, Medicare and Medicaid will stop reimbursement to hospitals who are having a certain amount of these cases pop up. If you're not taking care of your patients appropriately, Medicare and Medicaid will pull out funding. They won't support you anymore. That's a massive problem for hospitals, especially if you're a larger hospital. So adoption of universal precautions can also be included as far as actually controlling it at your own level, where you can look at it and maintain that your population of your employees are, keeping clean. So medical asepsis, surgical asepsis, and then the infection control officer. Medical asepsis, talking about hand washing, disinfecting surfaces, sanitizing things, washing soiled linens. All of that stuff counts, okay, as medical asepsis. Surgical asepsis, this is a little bit higher in, level as far as keeping things clean. We're trying to keep sterile conditions. There is a difference between something that has been cleaned and something that is sterile. I'm not going to get into the qualifications for what is sterile yet, because that is a chapter 11 thing that we'll get into when we get back from the exam, but we're going to talk about that later. So obviously surgical asepsis, you want more cleanliness involved, because we've got open bodies that should be sterile. You don't want to be introducing anything into that while that's open. The infection control officer is the one who implements these practices and procedures throughout the hospital. They will track outbreaks within the hospital, identify breaches in asepsis within the hospital, and then train any of the workers in the aseptic technique. So they are the ones that's actually in charge of everything that's going down in that hospital as far as keeping everything under control. So it's not going to be the doctors or the board members or the nurses or anything like that. These guys, the infection control officer, is the one that actually makes the rules that you will have to be following, what PPE you have to wear, when to change your gloves, when you need to be washing your hands. All of that is controlled by the infection control officer, not anybody else. Might have your charge nurse talking to you about it, but that isn't the one who came up with the rules. It's going to be your infection control officer. Next, we move on to the reproducibility rate. I don't like that word. And the case fatality rate. Okay, so R naught. This is the term that I was talking about. Okay, R naught, at least in the review, if you guys joined it. This is the reproduction rate of that virus or bacteria or protozoa, whatever it is, fungi. This is going to be determining how many susceptible people that aren't infected yet, right? So susceptible people. That one person who is infected will infect while they're infected. So if you are sick with COVID, how many people are you likely to infect during your entire course of infection? So that's R naught. And that's going to be different for everything, right? So each new pathogen that we're talking about will have a different R naught, how many people you're likely to infect. Some of them are very, very high. Some of them are not. I don't know the R naughts for measles, but I know it's very high. I know that COVID, depending on the strain, is like at 3 to 5, somewhere in there, depending on the strain. But yeah, so that's not super high, but still, we already know about how COVID can spread. So, all right, so it also does assume, however, that the exposed individuals, the susceptible people who are being exposed, that they don't have secondary immunity against the microbe. So anytime we're talking about R naught, we're talking about it in terms of nobody's been vaccinated from it, nobody has any immunity from having had it before. Okay, just assuming people who are completely susceptible to it. All right, case fatality rate, the number of people who die from the disease, divided by the number of people who are infected. That means literally, if you get infected, the likelihood that you're going to die from it. Okay, that's the case fatality rate. What are the odds if you get sick with rabies that you're going to die from it, that sort of stuff. And of course, this is going to be based on people who don't get treatment. So not everybody will be getting treatment. So you want to look at worst case scenario there. All right, so healthcare decisions are often based on the R naught and the case fatality rate, but they can vary based on geographic location, circumstances of an outbreak itself, and whatever variant or strain or whatever of the microbe we're looking at. If you have a very high R naught disease, if you want to control spread, then you're going to probably need vaccination in order to accomplish that. Here we have on the left, the deadliness, which is that case fatality rate. And on the right, we have the R naught, the communicability. So how much it can be spread. So let's start with the deadliness. We of course have like rabies and HIV and other diseases that would fall into that 75 to 100% range. So rabies, we know if you start having symptoms from that, 100% fatality. If you have HIV and you're not getting treatment from that, 100% fatality. There's other diseases that'll fall into the lower area. All right, then we have TB, Ebola, and plague. Notice how these are less deadly. Okay. Syphilis, next, kind of on its own. I don't know why, but there it is. And then these ones that are 0 to 24, rhinovirus, cholera even is down there. Smallpox is even down there. So these are even still some very serious diseases that even the government is still concerned about, but they're still down there in that bottom. And I don't know if you know this, but a 24% case fatality rate would still be very high loss of life. Most diseases aren't going to have that we encounter. All right, moving over to the communicability, how likely it is going to be spread around, right? Measles, malaria, mumps, rotavirus, and pertussis are extremely communicable. Malaria is not going to be spread from person to person, right? But it is going to be spread very easily by mosquitoes in those areas where it is endemic. So it's very likely you will get it if you are not on some sort of prophylactic medication. Very communicable, chickenpox, we kind of know that already. Things that are communicable just kind of at a normal level, we have smallpox, dengue fever, which is also mosquito-borne, tuberculosis, polio, rubella, and rhinovirus. Rhinovirus, just to be clear, in case you don't know, is just a cold virus, common cold. So pretty communicable. So is smallpox, yeah? I don't know if you know that, but smallpox, they used to just like have like scrapings of skin onto blankets and throw it to like, indigenous populations to try to wipe out populations to steal whatever their resources were. You don't know that about history. Surprise! Like that's what people used to do. The Spanish were very well known for that. Spanish were well known for that. Not that other populations didn't do it, but they definitely were well known for it. C. diff, cholera, Ebola, Lyme disease, norovirus, rabies. These are not considered super communicable, somewhat or minimally communicable, right? All righty. So it's very difficult to calculate R naught for microbes while a pandemic is actually in progress. So still in progress with COVID-19, we still have a lot of different strains out there. So it's kind of difficult to estimate that, but we can do the best we can. Early estimates were 2 to 5. Now we have the variants that we can actually separate out. Delta is between 3 to 5, and Omicron is starting at 5, maybe more. So it's a lot more communicable than the other ones. All right, vaccines. Vaccines play an important role in controlling infectious disease. Here we're talking about herd immunity. If you've ever heard the term of herd immunity, we're going to talk about exactly what it is and why it's beneficial and why it's something that you should strive for in a medical setting. So basically, you're going to be achieving herd immunity by making sure a certain percentage of the population is vaccinated with the vaccine for X disease, right? So usually vaccines are going to go through many layers of trials through experimental animals as well as through humans before they are licensed, which we'll talk about in just a second. But here we have herd immunity in the case of having the actual vaccine for coronavirus. Okay, so if you have COVID here, this person in orange has COVID. He's going to be our case that's exposing everybody else. Everybody in the purple is immunized. The people in the green are not immunized. They're healthy. They can get infected though. Okay, so now this person in orange that was up front, he's now naturally immune because he recovered. He didn't die from it. He was lucky enough that he didn't die from it. And then we have, he has natural protection from it. That's the point there. Then we have somebody back here who did get infected and might be a concern later on, but most of the people aren't getting infected here. And that's just because if enough people you're going out and being in contact with are vaccinated, they can't carry the disease to the next people, or you can't give it to them and that sort of stuff. So if you get enough people, a certain percentage of people vaccinated and covered based on the communicability of the disease, you can protect the entire population. That's what the idea is. It will take, so you can see down here at the bottom, same situation, but nobody has any immunity. This guy did survive it, so he has natural immunity. Now all these other people are sick, and some people are even dying. Look how many more people actually got sick from it, and some people even died this time. that's something to be aware of. Now, measles is something at least 95% of the population, might be even more than that. At least 95% of the population has to be vaccinated with MMR in order for herd immunity to exist. Okay. So that means 95% of the population actually got MMR vaccine. That means 5% of the population have a little bit of leeway, right? That means that's including little tiny babies that are just born, right? And elderly folks who have like a weakened immune system and can't be protected that well by vaccines anymore. Or people who are pregnant, maybe that can't get the vaccine while they're pregnant and didn't get it previously. Stuff like that. Those people can't get the vaccine. That's a different thing, right? Than people who choose not to get the vaccine. So you start adding in people who are choosing not to get the vaccine for whatever reason. But mostly, yes, of course, I am pointing fingers at anti-vaxxers. Those people are adding into that set of what the 95% can cover for. And in fact, maybe even stepping it back past that where you cannot achieve the 95%. And now you have outbreaks of measles in places like Florida schools. Like there was just a report of that just like less than a week ago. There's an outbreak of measles at Florida elementary school. That shouldn't be happening, right? I mean, I shouldn't have to say that though. That shouldn't be happening. It's, to me, irresponsible. This isn't about you. This is about everybody in the population and all the people that can't get the vaccines. It's not about you, right? If you can show me that you actually can prove the danger in these vaccines at a level that isn't worth protecting the rest of humanity, then come and talk to me. I'd love to hear the argument that you have to put forward because I don't believe you. It's just all I'm saying. Are there, of course, well, we're going to talk to it in a moment, but we'll get to it. We had the emergency use authorization in 2020, 2021 that allowed us to use the mRNA technology for the COVID vaccine, the one that got the Nobel Prize. So yes, they started phase 3 before phase 2 was concluded. They had most of the results that they needed. They didn't get all of them, so they went ahead and got approval to continue with phase 3 before that. About 100% of the vaccine side effects are usually going to be seen during the first 6 weeks after a vaccination and just in general, but especially for COVID, okay? But they also have a study of the effects of vaccines for their actual, side effects, and that would be called a phase 4 study, if that's happening. And they do watch that for COVID because they did roll out before they were completed with that other phase. Phase 2 wasn't complete yet. They had enough data. They were comfortable with moving forward though. And they did complete phase 2. They just started phase 3 before they completed it. So there is a vaccine adverse events reporting system. Gosh, I can't use it. Anyways, if you have problems with having gotten the vaccine, you can call this number or visit the website or whatever it is, or you can even contact your doctor about it. And anybody can report any adverse effects. It could be the patients themselves, it could be parents of patients, or it could be even the healthcare providers. They can report any of these negative effects of these vaccines so that they can keep track of what's going on where to monitor that sort of stuff. And there's also a vaccine injury compensation program where if there is some sort of injury sustained from the vaccine, in that case, we're talking about something that actually affected you either permanently or so seriously that you couldn't work or something like that, and it would cause some sort of monetary harm to you or loved ones, something like that, bodily harm that was permanent, that sort of thing. Usually in this situation, plaintiffs, the people who are complaining about whatever it is that they've sustained, do not have to pay court fees or attorney fees. That's covered by the government. All right, so right, I don't remember what I was going to say about the other thing unless I was going to get to it, but whatever. All right, emerging diseases and re-emerging diseases. An emerging disease is a new identified microbe. This does, however, include new strains of flu. Every time we have a new flu strain, if we have to roll out with a new vaccine, then that's a new disease. Essentially, it's an emerging disease. If it's new to your immune system, then that's an emerging disease. Okay, so that does count. When we had SARS come out back a long, long time ago, that was brand new disease to the population. So emerging disease. Re-emerging diseases, these are things that have affected the human population in the past that are coming back due to travel habitat invasion, or development of drug resistance. And probably more importantly, the drug resistance one, because that's something we can maybe potentially do something about. Not that we can't for travel and habitat invasion, but it makes more sense for what we do. All right. For monitoring the diseases, we have global disease detection and the CDC. I don't know what else to say about that. We have neglected parasitic infections. This is CDC trying to track parasitic infections, which remember, that's going to be protozoa or helminths, right? In the United States, tend to exist in poor populations and poor neighborhoods that are getting poor healthcare. And a lot of them are immigrants from other countries, maybe potentially even bringing in other diseases into these areas. So this, I know, looks like this is a gentleman out in, I don't know, sub-Saharan Africa or something, but this is talking about in the United States. I want to make that clear. All right. So here we have some neglected parasitic infections in the United States. Chagas disease caused by Trypanosoma cruzi. This is transmitted by the kissing bug bite. We have neurocysticercosis. This is the tapeworms, typical tapeworms. If you were to try to be one of those people that wants to lose weight and eat one of those prostaglandins, or what, I don't know what they're called, not prostaglandins, what are the proglottids, that's what they are. I always forget it. The proglottids that contain all the little eggs, those little pieces that break off of the tapeworms, you know, if you ingest one of those like a little pill, because you want to lose weight so your tapeworm can eat up all your calories, most of the time when you do that, it's not going to go into your gut. It's actually going to migrate into your brain. So that's neurocysticercosis. Is that some sort of thing that you shouldn't really be too concerned about? Well, if you're not swallowing tapeworms, then you're probably okay. But if you happen to get this disease, there isn't a whole lot they can do for you. The cysts that develop inside of the brain, that if they rupture, cause such a severe inflammatory reaction so quickly that the brain will cause brain damage to itself, like almost instantaneously if it ruptures. So there's not much they can do as far as like removing it. But you know, terrifying when you think about that part of it. Then toxocariasis, this is worms that travel through tissues and can cause blindness. You want to guess why they cause blindness? Because they go into your eyeballs, okay, eventually. Toxoplasmosis, this is the one with the cat litter we already talked about. Trichomoniasis is the disease caused by Trichomonas vaginalis, which we looked at in the eukaryote thing, right? That's going to be sexually transmitted protozoan disease. It can be leading to birth defects and premature births in mothers who have that. Also of concern, bioterrorism. This is anytime that we're trying to threaten, you know, the country basically by using microbes and whatever, so living organisms. So we did have anthrax in 2001. It was sent to Congress or whatever, and they had to deal with that. But yeah, you don't hear a whole lot about bioterrorism in the United States. Then we have agroterrorism, which is a terrifying concern, use of microorganisms to decimate agricultural industry. Now, of course, this is a concern for like crops and stuff like that, but specifically when I was working on my virus and at Penn, there was a girl in she was at my bench, like on the other side, that I worked with her because we had similar viruses. And she was studying Rift Valley fever virus. It's also a hemorrhagic fever virus. And it's BSL-3, so you can actually work with it in BSL-3, but it's 100% abortive in cattle, okay? So if cattle get sick with it and they have calves, they 100%, the calves will die or abort spontaneously. So that's not here in the United States. That's in the Rift Valley, which is in Kenya area in Africa. And so they do massive screening on any livestock that could be coming in to make sure they are not carrying Rift Valley fever virus. Because if that got into the United States, I mean, I'm not sure you understand how much of this nation depends on agriculture. Massively. I mean, it would destroy the country. It would easily. So you think the economy is bad now? Wait till you don't have cattle. So it's not all, oil and natural gas. So that would be a massive problem. I don't know if you've heard of Plum Island. That's where they do all the screening of like animals that are coming in from overseas, trying to make sure that they aren't, got disease. They also check like fruit and vegetables and stuff like that there too. That's why they're so strict about that. If you ever travel overseas, you're not supposed to be bringing stuff in. We do have the mosquitoes that can harbor that virus. It's just we don't have it here yet. So that's why they want to look out for it. So here is our little question of the chapter, which is what is the difference between a point source epidemic and a common source epidemic? And this, why does that distinction matter? Remember, point source, one person, everybody, well, one source, everybody getting sick all at that one time during that one exposure. And then the other one could be one source, but over time, like the cholera thing. Okay, so now we're going to do chapter 25. Or like I said, it's not going to be a lot. I'm probably not even going to go over all the slides, just what I care about. Okay. I would be aware of, kind of obvious one, the carbon cycle. That's the easy one, I feel like, the carbon cycle. Because what does it involve? It involves plants fixing carbon dioxide, inorganic carbon, into organic molecules, which we learned about with photosynthesis, right? And then the other animals after that, including saprobes and other heterotrophs, they're going to be utilizing those carbon, organic carbon molecules from the plants and exhaling it as waste, right? As carbon dioxide again, after breaking them off during Krebs cycle and all that. And exhaling it back into the atmosphere and then cycle through the plants and then into the animals and then cycle and so on and so forth. Of course, we have combustion as well, right? So it's going to be things like burning, volcanoes, industry, and all that. All right, so that's pretty easy. Cycling elements, whatever. I don't need to do too much of this. We did carbon. Yeah, so here's the picture of it if you're interested. So bringing that carbon dioxide in the atmosphere down into the plants for photosynthesis, they fix it into usable carbon, organic carbon that can be used by all sorts of organisms directly or indirectly, and then back carbon dioxide in the atmosphere. It's a lot simpler than it really looks, I feel like. Okay. This is the global monthly mean carbon dioxide amounts in the atmosphere since 1960. I used to have this little chart that showed the average temperature of the planet, like over time. And I mean, like it's undeniable, like warmer and warmer and warmer and warmer and warmer and warmer, like over time, like by far. So it's kind of terrifying when you think about that stuff. Does it have to do with carbon dioxide? Yes. Does it have to do with methane? Yes. Does it have to do with other things though? Yeah. So there's a lot of factors going in there, but just saying. All right. So methane is actually a more potent greenhouse gas than carbon dioxide. Okay. Yeah. All right. Next is the nitrogen cycle. There are 4 basic types of reactions in the nitrogen cycle. Okay. Nitrogen fixation, ammonification, nitrification, denitrification. There are chemical equations associated with these. Guess what? I don't care. So just know that those are the 4. And usually these are 3 that I will talk about because I don't want people to get confused with this as much. So just be aware of those are the terms, the correct terms for those reactions. Nitrogen fixation and other reactions with nitrogen are important because plants need to have their nitrogen in the form of ammonia, basically, in order to use it. All organisms do, or at least eukaryotes. So bacteria can do that for us as anaerobes and everything like that, turning whatever unusable nitrogen into a more usable ammonia form. In rhizobia, these bacteria will hang out in nodules in the roots of plants and give them nitrogen, basically. So that's pretty cool stuff. Okay, again, here's just descriptions of these that you don't need to know. Okay, just know the terms. I'm not going to ask about this stuff, really. There are organisms that live in the soil. They all work together. They have their own little ecosystem. That shouldn't surprise you too much. But yeah, there's all sorts of different organisms there. As you can tell, like this is going in more detail than you really are ever going to get tested on. Okay, aquatic microbiology. If you look at all of the water in the entire world and break it down basically into just what's the fresh water, what's drinkable water and everything like that, right? So down at the bottom, that dark blue with that little bit of light blue, that's dark blue is the saline based water and the light blue is the fresh water. Above that, we have ice caps, glaciers, groundwater, and stuff like that. You can't drink an ice cap or a glacier. I'm just saying. So anyways, put it in perspective. Most of our water is going to be coming from an aquifer. These are going to be surfacing through springs, geysers, hot vents. We all know that a lot of water comes from springs because you can go buy it at the store and it says springs, Ozark Springs or something like that. So aquifers are where the spring water comes from. Okay. There's a lot of organisms in the ocean, microbes too. We found a lot of new proteins and stuff like that in the ocean. The Dead Sea is an area that has very high saline content, salt content, so 35% salt, which is extremely high, but there are still microbes even there. In bacteriophages, we found there are 10 million viruses per milliliter in oceans. Now, 10 million viruses per milliliter. I don't know if you guys know this, but a milliliter is a very tiny amount, okay? It's a cc. If you guys worked in a hospital, then you know what a cc is. It's a cubic centimeter. So if that doesn't make it clear, let's literally make a cube out of centimeters. That's a milliliter. Okay. There are 10 million viruses, man. That's a lot. But yeah. So there's a lot of stuff going on in the ocean that has nothing to do with healthcare really as much. It's cool, but I don't know. Red tides. Okay. The plastosphere, I know that there's some sort of question about this on the test. It is plastic that's floating. It's waste plastic. They create sort of reefs where microorganisms can kind of build off of. It obstructs light in the ocean and puts makes a health risk for invertebrates and fish in the ocean, either from blocking the sun or just from the microbes that aren't supposed to be in that area, disrupting that the whole ecology of the area. Okay. Moving on to the freshwater communities, we know plankton are incredibly important for creating oxygen, basically. So plankton and cyanobacteria made most of the oxygen on the planet. I'm not going to ask you about the lake stuff, which is like oligotrophic and eutrophic. I'm not going to ask you about this. If you're interested in it, feel free to read about it. You won't be tested on it, okay? But what it has to deal with is oxygen being used up in the water, and then fish will die because there's not enough oxygen in the water for them to even survive, right? So that's the idea. Too much nutrients or too little nutrients can cause that sort of stuff. But let's move on to drinking water. Most water is going to come from aquifers, right? Rivers, aquifers, and springs. We can get it from in the ground or from the surface of the spring as well. So we're going to treat our water before it is going to get to your house. We'll have raw water that comes from wherever. Put it through coagulation, adding in a chemical to make particles stick together, make them larger so we can allow them to settle. And then we can filter out the water from what's settled to solids. We can do that several times. And then eventually we will filter the water through an actual filter and then treat with disinfectants, usually chlorine and or UV light to kill off whatever is in it. And then it'll go to your home or be stored somewhere for use, okay? So that's that water. In many parts of the world, your drinking water is also your sewage water, unfortunately. So we have wastewater draining out of homes and industries. That counts as sewage. We also have chemical debris and microorganisms associated with the sewage that can be a problem. And that also sewage in general, solid waste, dissolved organic matter, and toxic chemicals. So how do we treat wastewater? Okay, I don't know if you know this, but like always since you've been alive, they've been taking your poop water and cleaning it and putting it back into the environment. This is not news. It's not anything special or crazy or disgusting. It's neat. There's all sorts of steps involved in this to make it safe, put it back into the environment. And yes, they test it before they send it out. So primary stage, we're separating out the large matter. I probably don't have to explain to that what that really means, right? Physical things like plastic and stuff, but then, other things that aren't plastic. Next, we have the secondary stage. We're going to degrade everything else that didn't get removed in the primary stage, right? Some of this will be sludge, okay? So basically, it's the stuff that didn't get digested before, but it is not water. So we have bacteria that we can use to digest the sludge down. And so we can get as much water out of that as we can, because we want to conserve as much of the water as possible. We can even use some of the leftovers from all of this stuff for fertilizer and stuff like that, okay? Tertiary stage, now we're going to filter and chlorinate and UV just like we did with the other one. Now at that point, it's just water. We're just going to filter it a few times and everything. So here's our reclaimed water. We will use these for watering golf courses, watering at parks, releasing into large bodies of water as well. Okay, so we have to survey our water to see if it has any bad crap in it. So we did have to start monitoring COVID. When we realized that there was COVID in waste, they started having to figure out a way to filter it out. Luckily, they had a plan in place from poliovirus. Poliovirus is a much, much smaller virus. And it used to be filtered out from the water. We don't have poliovirus in the United States anymore, so they stopped worrying about that. So it's less expensive if they don't have to worry about it. But they just reinstate that in order to, treat your COVID so that we're not pumping COVID back into the environment, basically. So that's the idea there. So we do monitor for that. Good health is dependent on clean, potable water supply. That word, potable, it is pronounced potable, not potable. This is water that is free from pathogens. It's not necessarily clear. It doesn't necessarily taste good, but it's not got pathogens in it. Have you ever gone like camping or done Boy Scout stuff or anything that you like put iodine tablets in something and you can drink the water? It tastes terrible, but it is potable. It's not got pathogens in it. You do that. So free of pathogens. And you can, of course, filter it and still potable. It's whatever. But right, so prominent waterborne pathogens. We've already heard of some of these guys. Giardia, Cryptosporidium, Campylobacter, Salmonella, Shigella, Vibrio, as in cholera, Mycobacterium, hepatitis A, and Norwalk. All these are very concerning pathogens that we want to monitor in our water, make sure they're not there. So we have indicator bacteria, which are species that we have in the intestines of birds and mammals, that if they make it through the whole process of filtering and UV and all of that, if they're there in certain amounts, then probably the bad bacteria can be too, right? Even if you're not testing positive for them, the propensity is there for them to get through, okay? So we look for indicator bacteria. The ones that we focus the most on are our coliform bacteria, which are fecal bacteria that are non-harmful. So regular old E. coli, Citrobacter, Klebsiella, and Enterobacter. All of these guys are coliforms. So fecal coliforms are the ones that we're using as our indicator bacteria to tell us if we have the likelihood of pathogens making it through. Okay, so one way they can do this is the rapid plate count. The rapid method is the standard plate count where they're going to literally just plate the water samples onto media. Go figure, see if there's any colonies. They can also, if it's like water, and there shouldn't be very many organisms in it if you filtered it and all this, right? What they'll do is actually put a sample of water through a filter, and there's a little membrane that sits on there. You can take the membrane off and place that onto a medium and then see if anything grows on there. So it like concentrates them down. And so they'll try to figure out how many colonies will form from that. So that's the membrane filter membrane method. After filtration, put that filter onto a media. So you can count both non-fecal and fecal coliform colonies. So those are our indicators. This is kind of what I was talking about, just images of it. We did this for companies at my last job. Okay. So some fungi and bacteria are important for taste of your foods. Kombucha, if you don't know, if you haven't tried that. Pickled cabbage that is basically not always going to be sauerkraut, but sauerkraut's the best example you can think of for that. I don't want to try Norwegian fermented fish, thank you. All right, and then we have bread, of course. Leavening is achieved by Saccharomyces cerevisiae. That is like yeast for baking. It's not the same as yeast on your body, which is a whole different thing, Candida albicans. But these guys create compounds that give bread that smell that bread has. You know, it makes your mouth water just thinking about it probably. And there can be other microbes to add other flavors like Lactobacillus stampfrancisci that has to deal with sourdough. And of course, the production of beer. This is going to be creating ethanol and carbon dioxide, basically from a sugar, essentially, because they go through fermentation. Yeah. So how they're going to deal with that electrons? That's what they do. pyruvate plus electrons making ethanol. So this is all the steps in beer making, which I know literally nothing about this. Okay. This is absolutely not going to be on the test. I'll say the water coliforms, yes, water treatment, and then the stuff that we had mentioned previously about the cycles, carbon and the nitrogen cycles, that stuff, but not this, because I just don't know enough about this. I don't even understand. I could read these slides to you and they would not still not mean much to me. I should learn about it someday just so I can actually explain it, but I know that, I don't know, Dr. B is a lot better explaining this stuff than I am, but all right, and then that brings us to that concept of One Health, which it turns out apparently on the new textbook, you guys are lucky, there's a whole chapter for One Health, but it basically has to deal with keeping the whole planet healthy and how the health of some species has to deal with the health of other species and all of that stuff. Seems nice and happy and cheerful. Hantavirus. Oh, these are just new viruses and where they came from and talking about this. There's not much really to say about these, like hantavirus, which was transmitted by dried up urine and feces of mice. It does have a 50% to 70% mortality rate, which is very high. Okay, it is a hemorrhagic fever and it can cause kidney failure. That is found in Southwest United States. HIV, we already know, likely came from simian immunodeficiency virus. They're almost exactly the same. Which seem to have originated in Africa. We think from butchering primates. It's why we have Ebola as well. It's a similar concept. That's what they say. Came to the U.S. as GRID, which is gay-related immunodeficiency, because that's what they found at first. And of course, now they have learned better. Then other mosquito-borne diseases that are new to us being transmitted as the environment grows warmer, you know, more mosquitoes come further north. We have some mosquitoes, y'all. I live right by Overholser, and I mean like a street, like a neighborhood street from the actual lake. And so there's some mosquitoes over there, you guys, that are like massive, like massive mosquitoes already flying around. It started when it was even cold outside. I don't know how they do it. All right, SARS and COVID and where it came from, they think bats. Don't get me started on that. And then these are other new emerging diseases, recent diseases that have come out. I don't know anything about whatever this is. I don't know. I'm gonna have to look it up. I don't know what that is. And then re-emerging pathogens, things that are coming back out due to, you know, whatever, change in the environment and all sorts of things. These are the most concerning pathogens according to the World Health Organization. So worldwide, they're worrying about these. If you have had any loved ones lately who have had issues with, like I have my mother-in-law who she uses Humira. And so her immune system is lowered. And she started having like this yeast infection. I'm like, oh, it's going to be Candida albicans, but it's not. It's Candida auris, which I don't know much about, but apparently it's become really common nowadays to have that. That's a learning moment for me. That Crimean Congo hemorrhagic fever, that's what I used to study. All right, so that is that for chapter 25 and what we weren't able to cover last time. Monitoring indicator bacteria like coliforms, just be aware of why we do that. All right. Shall we knock out do you guys want to do 1 or 2 Kahoots if we have time? You want to do 14 or do you want to do, let's do 14. I feel like that's more beneficial, yeah. Lab is going to be fast and easy today, I promise. I mean it, genuinely. I think it's pretty much everybody. I'm going to go ahead and start it. so hot. I don't know why I didn't turn the fan on. Thought about it and I didn't do it. All right. Who was Jon Snow? What did he do? All right. Yep. He was involved in that, stopping that cholera outbreak at that well, water pump. All right. Which term refers to the total number of existing cases with respect to the entire population? All right. So this one is prevalence on this one. What is this doing? Which type of outbreak occurs when pathogens come from a single source and all victims are exposed at the same time? All right, good job. Point source. All right, true or false? A pandemic is the spread of an epidemic across continents. Excellent. Good job, guys. Which term refers to a disease that exhibits a steady frequency over a long period in a particular place? Okay. Yep, endemic. Think of endemics being like endo, so inside, so it's where you are. Okay, what aspect of a test is determined by how many positives were incorrectly tested as negatives? It's determined by how many false negatives there are. You want to reduce the false negatives. All right, good job, guys. That is sensitivity. Yes. Getting it. All right, what are Koch's postulates used for? We had this one in the last chapter too. Right, so Koch's postulates are used to determine whatever organism caused a disease. Two factors, exposure and disease, let's say, are collected at a single time with no reference to which might have occurred first. This is the one. This is the one I don't ever remember. Okay, yeah, so this is cross-sectional. It's like taking kind of a slice out of it and looking at just that one, those two factors, I guess. I mean, okay, we didn't study Hill's criteria, okay? We aren't doing that one. FYI, cut that out of our lecture. Sorry. But surprise, if you guys want to try to guess it, you can. The answers sound exactly the same to me. Okay, so it ends up happening, just so you know, to be the red and the yellow. It doesn't matter what it means because I'm not gonna test you on it. Forgot to take it out of there. What term refers to how many susceptible people on average that one infected person will spread the infection to. All right, so that is correct. That's R naught. All right, so get your pictures, then we'll move over to the lab.